Biomolecules. Now, by now we know that bio means living, molecules means all the molecular compounds which are present, may it be micro, may it be macro, which all constitutes the living organisms. We call them biomolecules. How to find out their chemical composition, right? So this becomes our first point of study, that chemical composition, where chemical composition of organisms as well as Earth's crust. This much we are aware that whenever the chemical analysis of any living tissue it is taken up, it shows the list of elements. Which list of elements? Carbon, oxygen, hydrogen, nitrogen, calcium, right? So many are there. But in what amount? The similar analysis is found in the sample of Earth as well. Sample of Earth's crust. Over there we have found that the same elements are present. Almost all are seen in the living tissue and they are in the earth crust as well. So very close examination, it reveals that the relative abundance of carbon and hydrogen. Carbon and hydrogen in comparison to other elements is much higher. Where? In the living tissue. Why? What's the reason? Obviously, whether we are talking about carbohydrates, proteins, fats, metabolites, all these are made up of carbon and hydrogen. So simply, when we talk about this, we say analysis. Analysis is something which tells us about why this chemical composition is higher. So analysis is done. What for? Of chemical composition of living organisms, right? When these are generally analyzed, we got to know that they are made up of organic compounds like proteins, carbohydrates, fats, lipids, fats or lipids, whatever we say, as well as inorganic compounds like calcium carbonate and sodium chloride. Fine. It's not only this, along with they have elements like potassium, sodium, magnesium, calcium, etc. All these are also present. In analysis, we came to know this. And we should not forget that water is the most abundant chemical where all these are present. Okay? Water is the most abundant chemical compound present in the body of all the living organisms. Then also all these constituents are present. Clear? Now coming to the chemical analysis. This is simply an idea. How do we get to know? How do we obtain all these carbon, hydrogen, oxygen, and how do we get to know their exact percentage? So we do the chemical analysis. Of organic compounds. See, to do this, we take a known weight of living tissue. Why a known weight? Because we will get to know how much amount of living matter have we taken for this chemical analysis and how much is received back in terms of different chemicals. May it be any fruit. We can take a fruit or a piece of liver of any animal. Then the second step is grind it. What do? We need water for grinding, but here we are not using water. We are using trichloroacetic acid, okay, which is this in the chemical formula. We use trichloroacetic acid. We grind it in pestle and mortar. Pestle mortar I'll show you in lab. Basically pestle and mortar घर में भी होता है. This thick slurry, what do we obtain? We obtain thick slurry. It is obtained, it is filtered. Then we filter it. Filtered how? Filtered through a cheesecloth. We need to obtain it in such a manner that इसके दो parts आएंगे. एक part आएगा filtrate, that is called the acid soluble fraction. ठीक है? The one which is collected here in the jar. Supposingly we are collecting it, तो वो जो collect होगा यहाँ पे, that is the filtrate. And the second one is retentate. It is known as the acid insoluble fraction. Right? This basically which is collected in the funnel here where we have used the cheesecloth. So this part is here where we have used the cheesecloth, and there only the filtrate will move. Right? This retent extract can be subjected to various separation techniques now to obtain a compound from all the other compounds. Now, when analytical techniques are applied, we get an idea about the molecular formula and the structure of the compound which is present here or here. This is one method. Clear? Second method. Second method is ash analysis. Ash analysis, this is basically done for inorganic compounds and elements. Right? We say one has a weight and a small amount of living tissue. Now we are taking here again a small amount of living tissue. That living tissue could be again a piece of liver, measured, a fruit and or a leaf. Right? We will now dry it. Dry it till the water evaporates. Okay, like the leaves, they get dried up when these are left in the garden. So entirely we need to dry it up till all the water in it evaporates. Okay, We took the initial weight. We have taken a small tissue, liver, fruit and leaf. The procedure will remain the same. We have to weigh it initially. And the initial weight, in this sense, that we have taken earlier, that initial weight, उसको हम बोलते हैं fresh weight or the wet weight. And the final weight, that will be known as the dry weight. So इसके बीच में तो हमें क्या डिफरेंस पता चल गया? अमाउंट ऑफ वाटर लेवल. क्लियर? नेक्स्ट, now when this tissue obtained after drying, this one now burnt completely. Okay, so what will happen? Carbon compounds will automatically get oxidized. To what? To CO2 and to water vapors. And whatever is left behind, we call it ash. This is one part which is happening. Number two, the left behind or remaining content is known as ash. Now this ash, it contains inorganic compounds like sulfate, chlorides, phosphates and elements. Which are elements? Elements like sodium, elements like potassium, calcium and magnesium. Clear? Now moving to, after doing these tests, we are moving to the biomolecules of the cell. Like all the carbon compounds which are found in the living tissues are known as biomolecules. Simply we say carbon compounds, right? Biomolecules of cells. So we say all the carbon compounds which are found in the living tissues are called biomolecules, right? These biomolecules whose weight is less than 1000 Dalton are called micromolecules, and those whose weight is more than 'whether they have more than this or they are with this molecular weight of 1000 Dalton, they are considered as macromolecules. We have got the acid soluble fraction which contains the organic molecules, right? Another thing which we need to know is acid soluble fraction which contained all the organic molecules. These are the organic micromolecules. Okay, and these are sugar, amino acids, nucleotides, and along with that some inorganic ones also. And those inorganic ones are sulfate and phosphate. Because if we are saying nucleotides are there, then sulfates and phosphates have to be there. Okay, then acid insoluble fraction. For that, it contains macromolecules. We call them proteins, polysaccharides, nucleic acids, and also lipids. You know, lipids are actually micromolecules. They are found in the acid insoluble fraction. That's why they are called. But found in acid insoluble fraction. That's why they are considered macromolecules. And the acid soluble fraction, it represents roughly the cytoplasmic composition of micromolecules. This one represents the cytoplasm, cell organelles, and the entire chemical composition is here in these micro and macro molecules. Right. Now coming to these components one by one. We have done that there are sugars, there are amino acids, there are nucleotides. Let's come to the structures one after the other. That what is the basic anatomy and the anatomical structures representing all these. Moving to the first structure, which is sugar. It is the simplest among the sugars, which we call carbohydrates. So simple carbohydrate. And if we say simplest, they are the monosaccharides. Mono means single unit. Monosaccharides. When we are saying monosaccharides, you have to remember this thing that they cannot be hydrolyzed further. इसको आप इससे ज़्यादा नहीं तोड़ सकते. Hydrolyzed further. This is the single unit of carbohydrates. They may be composed of 3 to 7 carbons. Okay. Carbon atoms. Now come to the structure. This one, this one, this one, this one, this one, these all are carbons. So here it is HOH, HOH, here OH, here. I'll write it with a different pen to make it appear differently. ये देखो. Right? On the other side. This structural difference will bring a little change in its chemical composition. CH2OH. If we have to count the number of carbon bonds, carbon atoms, how many are they? 1, 2, 3, 4, 5 and 6th carbon. So this is glucose, a 6-carbon compound. Fine. Moving to another one. Here I am showing HOH, 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 CH2OH. Okay. Now let's count the number of carbon atoms. 1, 2, 3, 4 and 5. It's ribose. When we say deoxyribose, then this OH group is replaced by H. That is called deoxyribose. Alright, it's a 5-carbon compound. Now, these monosaccharides, they have either a free aldehyde or a ketone group which can reduce the copper ions to form cupric ions, and they are called reducing sugars. I'll write it down over here. The monosaccharides have either a free aldehyde or ketone group which can reduce copper ions, hence they are called reducing sugars. Right? If I say disaccharides means sugars which have two monomers, two of glucose which are linked together. So examples, remember in digestion chapter we have done example of sucrose, maltose and lactose. These are disaccharides. Alright, moving to amino acids. The second organic compound that has been identified here, basically amino acids, ये कैसे बनते हैं? These are the organic compounds which contain an amino group. Organic compounds containing an amino group, then an acidic group, and substituents of the same. Basically these two are substituents on alpha carbon. Okay, and they are substituted methanes. We have to remember this. Amino acids are substituted methane, right, with its 4 substituent groups, with its 4 substituent groups occupying the 4 valency positions of alpha carbon, the first carbon, primary carbon, and these 4 groups that are hydrogen, carboxyl group, amino group, and a variable group which is designed as R group. That means the alkyl group, which tells us about the number of carbon chains over there. Okay. Now general formula that is derived here is, we know this is it, ये मीथेन होता है, ठीक है. This is substituted by COOH group. This is by NH2 group. This is by side chain. So what it looks like and what it becomes, it will be like this. Okay, now we call it an amino acid. Depending on this R group, there are 20 different amino acids that occur in proteins. Okay, I'm writing it down. Depending upon the alkyl group, okay, there are 20 different amino acids that occur in proteins. But we need to remember it that in case of glycine, this is the hydroxy group. This is, let's do it quickly. In glycine, this R group is hydrogen. And in alanine, this is a methyl group. And in case of serine, it is CH2OH. Okay, this is in serine. This is an example of amino acids. All right, glycine, alanine, and serine. So this R group is hydrogen in case of glycine, a methyl group in case of alanine, and a hydroxymethyl in case of serine. Methyl and hydrogen. And depending upon the number of amino acids, okay, depending upon the number of amino acids, it may be basic amino acids, acidic amino acids, and neutral amino acids, or aromatic amino acids. Okay, and a specific characteristic of amino acid is the ionizable nature. Basic, acidic, neutral, and aromatic. And a specific characteristic property is the ionizable nature. What is that ionizable nature? That is formation of ionizable nature of NH2 as well as COOH group, so that the structure changes in the solutions of different pH, to make sure that the structure changes in the different solutions of different pH. Alright, this is the ionizable nature. And you know, each amino acid is given a 3-letter abbreviation and a single letter code. Each amino acid is given a 3-letter abbreviation and a single letter code.